Preparation model paper 12 regarding ASRB net plant pathology. I manage Shankar Reddy, PhD plant pathology. First one is the term biological control was given by. So the questions completely related to biocontrol only, especially biocontrol section. The term biocontrol was given by Van Tubeff, Weinling, Valentine Relay, and Sanford. So generally the term biocontrol or the biological control was actually given by Van Tubeff. Next one, who suggested the biocontrol properties of trichoderma for the first time? Millard, Taylor, Weinling, S.D. Garrett. So generally this Weinling in 1931 and 1932 who first suggested the biocontrol properties of Trichoderma viridi on various soil-borne pathogens like Sclerotian ralphs, Rhizoctonia solani. He have done various kinds of laboratory tests and all those things, and he came to a conclusion that this Trichoderma can act as a biocontrol agent. Generally this S.D. Garrett we considered as a father of biocontrol agent when it comes to plant pathology. But in entomology, it is mentioned that Valentine relay is considered as a biocontrol agent. For them, that is separate. For us, SD Garrett is generally considered as a father of biocontrol agents or biological control. Next one, whose work is mainly related to wheat take-all disease? Sanford, Brown, Magron, and SD Garrett. So out of all the scientists, S.D. Garrett is generally considered as father of biocontrol agents or biological control agents. Almost many years until his death, he worked on especially a disease called wheat take-all disease, which is caused by Gaumonomyces graminis triticale. And he came to a conclusion that this is the disease. With the help of biocontrol agents, we can completely control the disease. And it is also mentioned in various literatures that there is only one disease that we can bring into the control 100% by using this biocontrol agents. Then Pseudomonas species can be used for this wheat take-all disease. So the scientist who especially worked on this wheat take-all disease, S.D. Garrett. In rhizosphere, rhiza means shoot, root, branches, leaves. So in rhizosphere, rhiza means root. Rhizobacteria means that is a bacteria which are present near the root region, that is called rhizobacteria. We can take an example of PGPR, plant growth promoting rhizobacteria, something like that, right? So rhiza means generally root or hair-like structure. Generally that is root also look like hair-like structure, right? So rhiza means root. The next one is which one is used as a biocontrol agents against the plant diseases? So Trichoderma viridi, Glomus, Bacillus thuringiensis, baculoviruses. So generally among all the given options, Trichoderma is generally considered as biocontrol agents. For example, if you see this glomus is like VAM, this vesicular arbuscular mycorrhiza fungus, something like that it will comes under. But Bacillus thuringiensis is BT cotton. In BT cotton, we use a special kind of what can we say that genes, so that BT genes it contains, right? So cry genes or cry A, cry B, something like the genes will be present. This is Bacillus thuringiensis. Baculoviruses are the viruses which act as the viral biocontrol agent, we can say. But generally the biocontrol terminology, this is among the given options, it is related to Trichoderma. So which of the following biocontrol agent which is used for the nematodes? Pseudomonas sapiaceae, Pisolithus tinctorius, Phacillomyces liliaceanum, Gliogladium virens. So among the given options, Phacillomyces liliaceanum is especially recommended for against various nematode diseases, especially in plants. So the answer is Phacillomyces liliaceanus. The second one is a biocontrol agent used against grapes powdery mildew, not a downy mildew, it's a grapes powdery mildew. Candida oleophila, Trichoderma harzianum, Amphilomyces quiscalis, Coniotheri minitans. So this Amphilomyces quiscalis generally are especially recommended for grapes powdery mildew disease. So for grapes downy mildew, we do have a fungicide called Bodomycin, right? For grapes powdery mildew, we can recommend this Amphilomyces quiscalis. We can also call it as AQ10, otherwise called as Amphilomyces quiscalis, especially recommended biocontrol agents for this grapes powdery mildew. The next one is a biocontrol agent used for crown gall diseases, crown gall of apple, crown gall of pear, generally crown gall of stone fruits. All the crown galls are mostly caused by Agrobacterium tumefaciens. This Agrobacterium tumefaciens, now called as Rhizobium radiobacter based on the 16 srRNA. So now the name is changed, but still this Agrobacterium tumefaciens is still we are using in literature. So the biocontrol agent recommended is the Candida oleophila, Pythium oleandrum, Glyocladium catenolatum, and the next one, last one is Agrobacterium radiobacter. Generally, there is a strain Agrobacterium radiobacter. There is a two strains are available in this Agrobacterium radiobacter. Agrobacterium radiobacter K84. Agrobacterium radiobacter K1026. There are the two strains which are available for the management of this crown gall diseases. So both are Agrobacterium, right? Then what is the difference between Agrobacterium tumefaciens and Agrobacterium radiobacter? This Agrobacterium tumefaciens contains a TA plasmid, that is a tumor inducing plasmid, which act as a pathogenicity factor, where this Agrobacterium is lacking of this TA plasmid, hence it is not pathogenic. So that's why we are using as a biocontrol agent and exploiting against this for the management of this crown gall diseases. I repeat once again, crown gall of apple is caused by Agrobacterium tumefaciens. For the management, there is a bacteria called Agrobacterium radiobacter. Two strains are available, Agrobacterium radiobacter K84 1026 with the trade name of Digall and Nogal. Okay, so these are especially recommended for the management of this crown gall diseases, crown gall of stone fruits generally we can call. The next one is the biocontrol agent used for buttrot of pines, Candida oleophila, Penifora gigantea, Plebiopsis gigantea, both B and C. So generally the first used biocontrol agent or first biocontrol agent is a Penifora gigantea. The Penifora gigantea is also known as Plebiopsis gigantea. So the answer is both B and C. The first biocontrol agent which is exploited against this buttrot of apple is Heterobasidium annosum. This buttrot of pines, sorry, not apple, buttrot of pines is generally caused by Heterobasidium annosum. So this for the management of this Heterobasidium annosum, the first biocontrol agent recommended is Penifora gigantea or Plebiopsis gigantea. The next one is a biocontrol agent for rust, Ampeliomyces quisqualis, Darluca Candida oleophila, Fusarium sambucinum. So the rust diseases, for the management of rust diseases, the best recommended biocontrol agent is Darluca. So we already discussed this Ampeliomyces quisqualis is generally recommended for powdery mildew of grapes. This Candida oleophila is generally referred for Venturia kind of disease, like maybe apple scab, which is caused by Venturia anaequalis. I think I'm so. Uh, sorry. This is Venturia anaequalis causes apple scab only. The next one is Arctic nets are found in ectomycorrhiza. General ectomycorrhiza produces this Arctic nets. Then wheat take-all disease is caused by, I think I already discussed this, wheat take-all disease is caused by Gaumonomyces graminis tritici. So Vaccinia graminis tritici, wheat stem rust, Hardium vulgare, that is a barley rust. The next one is a biocontrol agent recommended for Venturia inequalis. I think we have already discussed, right? Venturia inequalis, which causes apple scab. So the first plant bacterial disease is a fire blight of apple, which is caused by Irvinia amylover. That is a bacterial disease. Okay. So Ampeliomyces quisqualis, it is normally for powdery mildew. Fusarium oxysporonot, Coniotherium minutus, Candida oleophila. Generally, this Candida oleophila, I think we already discussed it. Candida oleophila generally recommended for this Venturia inequalis, that is apple scab. The next one is the trade name of Agrobacterium radiobacter K84. I think in the previous slide only we discussed it, right? So Galtrol, Nogal, Digall, Mycostop. The trade name of K84 is Galtrol. 1024 is the Nogal. Okay. The next one is the source of the antibiotic gliotoxin. So the gliotoxin is the which bacteria it produces gliotoxin? Bacillus, Pseudomonas, Agrobacterium, and Trichoderma. So this Trichoderma generally produces various kinds of antibiotics like trichodermine, viridin, gliotoxin, all the toxin kind of substances which are produced by this Trichoderma for the suppression of the diseases. So this gliotoxin is generally produced by this Trichoderma viridi. Phenylzine 1-carboxylic acid plays an important role in suppressing the wheat take-all disease is actually produced by. So this is the phenylzine 1-carboxylic acid. This is the compound. so which is actually plays a very important role in the management of wheat take call disease that is produced by. So I already told you wheat take call disease is the only one disease that can be effectively managed 100% by using a biocontrol agent called Pseudomonas, right? So here this phenanzarin is actually produced by Pseudomonas fluorescens. Among the given options, Trichoderma viride, Bacillus subtilis, this Pseudomonas fluorescens is effectively used for the management of this wheat take call disease, that is Gommonomyces graminis triticale. The next one is a selective medium for the isolation of Trichoderma from soil samples was developed by Kings et al., Eladchet, Windling, Seabooth. So here in people, while you guys are writing thesis, you might have encountered with this Trichoderma viride selective medium which was given by Elad and Chet 1983, right? So this is a very very important one. The Trichoderma selective medium, TSM, we can also call this TSM, Trichoderma selective medium. So this Trichoderma selective medium was given by Elad and Chet. Kingsby medium, which was given by Kingsby medium, generally recommended for the specific medium for Pseudomonas. This is Seabooth wrote monograph on Fusarium. Siderophore produced by Pseudomonas aeruginosa, bacitracin, pyoveridin, deferoxamine, and Fusarium C. So this siderophore actually produced by the Pseudomonas aeruginosa is pyoveridin is the siderophore which is produced by Pseudomonas aeruginosa. And the next one is the dual culture technique for testing of biocontrol agent efficacy was developed by Dennis Webster, both A and B reddish. Actually answer is both A and B, because the dual culture technique was given by Dennis and Webster in 1971, I think. So the dual culture technique was given by Dennis and Webster. The next one is the trade name of Agrobacterium tumefaciens. So tumefaciens, they are asking, they are not asking Agrobacterium radiobacter, they are asking Agrobacterium tumefaciens. So probably Agrobacterium radiobacter it's supposed to be, maybe let's see, quantum 400, Galtrol, F-staph, Gliogard. Actually this is not related to the options given, probably there might be a mistake. So since we have already seen K84, right, Agrobacterium radiobacter, let's see here Agrobacterium radiobacter 1026. So that is K1026. There are two strains are available that I told you, Agrobacterium radiobacter K84, Agrobacterium radiobacter K1026. K84 generally the trade name is Galtrol. This radiobacter K1026 is Nogal, N-O-G-A-L-L. The next one is trade name of Pseudomonas fluorescens, Daggergy, AQ10, Binabti, Mycostop. So these are all mycoherbicide kind of biocontrol agents. These biocontrol agents are especially recommended for management of herbs like herbicides, weeds, right? So the Pseudomonas fluorescens generally sold outside with the trade name of Daggergy. Active ingredient of neem, azadirachtin, nimbidin, all the above actually. So this neem contains azadirachtin, indica contains azadirachtin, nimbidin, all the ingredients which are present in this neem. The next one is the concentration of AVP. AVP stands for antiviral principle used in the management of plant diseases. 1%, 5%, 10, 20. So in the AVP preparation also, the sorghum leaves or coconut leaves can be used at the concentration of 10%. So at the end, sorry, so the output solution can be recommended at the percentage of 10%. So which of the following are used as a AVP, antiviral principle, for the management of generally plant diseases? Okay, so Prosophis juliflora, Cyanodon dactylan, sorghum leaves, all the above. So the option answer is all the above, because there are some list of plants or plant leaves can be used for the preparation of AVP. The first one is coconut leaves, sorghum leaves, Prosophis juliflora, Cyanodon dactylan, and bougainvillea. These are all the various kinds of plants or plant leaves or plant products can be used for the management of antiviral principle. So this antiviral principles are especially recommended for viral diseases like groundnut bud necrosis virus, tomato spotted wilt virus. So such kind of diseases we can effectively use this antiviral principles. So generally antiviral principles are extracted from sorghum leaves or coconut leaves, Prosophis juliflora, Cyanodon dactylan, and bougainvillea as well. So the answer is all the above. So the trade name of azadirachtin Neem Mark, Neem Gold, Nimba, Veligo. So generally other trade names also available, but the most widely selling trade name is a Neem Mark. Neem Mark is the trade name. So Coligo is a mycoherbicide. Mycoherbicide means which is used to kill the herb, right? So which is extracted from fungi. So myco means fungi, herbicide means you guys know which is used to kill the herbs, that is especially herbs in the sense weeds. So Coligo is a mycoherbicide which is extracted from Coltotrichum gliosporides, Phytophthora palmivora, Alternaria cassi, aldabo. So the name itself starts with Coligo, Coltotrichum gliosporides. So Coltotrichum gliosporides, there is a fungi which is actually generally causes anthracnose, right? So from that pathogen they isolated a compound or mycoherbicide called Coligo. Which of the following is an entomopathogenic fungi? Bavaria bassiana, Pseudomonas fluorescens, Trichoderma harzianum, all the above. So Trichoderma harzianum and Pseudomonas fluorescens are the biocontrol agents. They are not entomopathogens. They are not pathogenic on insects, entomology insect, right? So this Bavaria bassiana is especially highly used for the management of insect. This Bavaria bassiana and one more entomopathogenic fungi, Verticillium lacani. And the fungal hyperparasites are also called as, so fungal other name of fungal hypoparasites, mycoparasite, fungal predators, mycophages, all the above. So generally this fungal hypoparasites are called as mycoparasites. So the antibiotic derived from Pseudomonas fluorescens, gliotoxin, herbicolin, 2,4-diacetofluoroglucinol, phenenzanes. Generally, this Pseudomonas fluorescens produces an antibiotic called 2,4-diacetofluoroglucinol. Of course, this phenenzane is also the compound that is produced by Pseudomonas fluorescens. So, among the given options, this is more closely related to this question, 2,4-diacetylfluoroglucinol. The next one is the first biocontrol agents, Penniphora gigantea, as we discussed, right? Penniphora gigantea, Plebiopsis gigantea, which is especially used against the bud rot of pines, which is caused by Heterobasidium annosum. So which was first obtained or produced in the year 73, 63, 83, 1993. So generally the first obtained in the year 1963. The next one is the first biocontrol agent, Pseudomonas fluorescens, Penniphora gigantea, Trichoderma mobility. Just now we discussed, right? First biocontrol agent, Plebiopsis gigantea or Penniphora gigantea. The trade name of Agrobacterium radiobacter K1026, Nogal, we have discussed, right? The next one, the trade name of Bacillus subtilis GB03, Nogal, Campaign, Kodiak, Aspire. So generally this Kodiak is the trade name that is recommended and they are selling the Bacillus subtilis GB03. Genome of hypovirulent strains used in the hypovirulence phenomenon. So the first of all, so in the theoretical section I have explained all these things, what is hypovirulence and all those things. So since the exam is approaching very fast, we are planned this MCQ kind of things. So before seeing this MCQ, we also suggest you to see the videos that we have published in the previous years. We have published a couple of videos around maybe 20 or 25 videos we have published in the previous year for preparation of net. So first see that videos, then come to this MCQ kind of stuff. So that will be very easy. So if you have a knowledge only, then only you can throw your overall broad spectrum knowledge on it. Otherwise it will be just like a mug up, right? So it won't be useful that much. So I suggest you to watch the videos first, then come to this kind of things. Anyway, I will tell you what is hypovirulence. So just reducing the virulence of the pathogens by using some kind of pathogens. So this pathogens, what they're asking is this strains of pathogens, that viral pathogens, which contains what kind of DNA they're asking. Double-stranded RNA, single-stranded RNA, single-stranded DNA, double-stranded DNA. So this double-stranded RNA pathogens, which is used for this hypovirulence phenomena, which is used to reduce the virulence of the pathogens. Generally, this hypovirulence phenomena can be recommended for a disease called chestnut blight, Cryphonecteria parasitica. The next the chestnut blight, yes, the name itself, the next question itself is a chestnut blight only. Chestnut blight controlled naturally by using the hypovirulent strains in Italy, France, Europe, none of the above. So this hypovirulence phenomena, especially Italian people are they used to grow lots of chestnut trees for various kinds of purposes, and natural burning fuels and all those things, they use this chestnut. So Italy people can use more, and couple of France, Europe people as well, but Italy is more famous for that. And chestnut blight controlled artificially by using hypovirulent strains. Again, the same question was repeated. I don't know why. Probably there will be small misunderstanding. So anyway, Italy, France, Europe, and all the answers it will come. So the answer comes to Italy and as well as France as well. The next one, which one of the following is a fungal biocontrol agent? AQ10, Pseudomon, Dene, Kodiak. So this AQ10, Amphiomyces quisqualis 10, is a fungal biocontrol agent. Remaining all are bacterial biocontrol agents. Which biocontrol agent is used for protecting the lemon from Penicillium green mold? Generally, if you see the citrus fruits, we can see some kind of moldy growth if we kept for a couple of days, right? So that is generally Penicillium. For the management of this disease, so what is the biocontrol agent used? AQ10, Canidomonas oleifera, Coniotherium minitans, and Pseudomonas syringae. So generally, Pseudomonas syringae can be recommended for the management of this disease. The next one is the biocontrol agents for coronation rust: Darluca phylum, Verticillium lacani, AQ10, all the above. So Verticillium lacani can be especially recommended for this coronation rust. The next one, fire blight of apple can be controlled by the spraying of Irvinia herbicola, Irvinia amylovora, Bacillus subtilis, Pseudomonas fluorescens. So this Irvinia amylovora, which is responsible for the fire blight of apple, right? So for the management, there is a this Herbinia herbicola generally recommended for the management. The next one is, which biocontrol agent is used for the control of wood decaying fungi? Trichoderma harzianum, AQ10, Myorrhizium verrucaria, and Coniotherium minitans. So generally this Trichoderma harzianum can be recommended for the management of this wood decaying fungi. The next one is a biocontrol agent recommended for Sclerotiana sclerotiorum. This is white fungi. Generally we can call it as what we can call it as white mold fungi or something. So Trichoderma harzianum, AQ10, Myorrhizium verrucaria, and Coniotherium minitans. Here Coniotherium minitans is especially recommended for against this sclerotium kind of disease, especially Sclerotiana sclerotiorum. The next one is a biocontrol agent for groundnut thicka disease, early thicka, late thicka. There are two kinds of thicka diseases are there, right? Cercospora arachidicola, Cercospora personatum, or Mycospora arachidicola, Mycospora berkeley, something like that will be there, right? So the biocontrol agent recommended for this thicka disease is Diciama pulvinata, Verticillium lacani, Darluca phylum, both A and B. Generally both A and B can be recommended. This Verticillium lacani and Diciama pulvinata can be recommended for this early thicka disease. Biocontrol agents for Womycetes group of fungi, almost all Womycetes group of fungi. Chaetomium globosum, Glycladium virens, Candida oleophila, Fusarium sambucinum. This Chaetomium globosum is especially recommended for almost all Womycetes group of fungi, and it is very effective against this Womycetes group of fungi. The next one is a biocontrol agent recommended for cucurbits powdery mildew, AQ10, Coniotherium minitans, Teleopsis, both A and B. So generally Coniotherium minitans and AQ10 both can be recommended for powdery mildew. Generally grapes powdery mildew, right? Not only for grapes powdery mildew, all kinds of powdery mildews we can recommend this AQ10. Along with Coniotherium minitans is also effective against this cucurbits powdery mildew kind of diseases. Biocontrol agent for root knot nematode: Pasturia penetrans, Bacillus penetrans, Fusarium sambucinum, both A and B. Again same, both A and B. Pasturia penetrans and Bacillus penetrans can be recommended for this root knot nematode. Biocontrol agent for ergot disease: Strigula elegans, Candida oleophila, Fusarium sambucinum, Verticillium lacani. Fusarium sambucinum is especially the biocontrol agent recommended for ergot disease, all kinds of ergot disease, Fusarium sambucinum. So biocontrol agent for coffee rust: Verticillium lacani, Verticillium hemilia, Coniotherium minitans, both A and B. Both A and B. Verticillium lacani and this Verticillium hemilia can be recommended. Biocontrol agent for red rust: again same, both B and C here. Strigula elegans and Strigula aesthetica can be recommended. The last one is a biocontrol agent for citrus gummosis. So Chaetomium globosum, Trichoderma aspergillum, Candida oleophila. So Cutomium globosum and Trichoderma asperellum both are recommended for this citrus gummosis. Generally, this citrus gummosis is caused by 6 species of Phytophthora: Citropthora, Phytophthora, Palmivora, Phytophthora parasitica, Phytophthora, various ricci, some kind of species are there. Almost 6 species of Phytophthora can be recommended for this citrus gummosis. These are all the answers. There might be one or two mistakes are found in, especially in the question number 35 and 36. So the both questions are same, but the answers given here is wrong. In Italy and as well as France, both this hypovirulent can be used. There might be a little bit mistakes found in. Please, if you guys found the mistakes, please bring it to our notice. We will rectify it in the further editions. For further more information, students can refer this book, A Vision into Plant Pathology, complete student version. For further guidance and clarifying the doubts, a student can reach us at www.geekyresearcher.com. Stay geeky and stay tuned. We are Team Geeky Researchers.